All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar is brought to you today by Plantae, the open online community for plant sciences powered by the American Society of Plant Biologists. Next slide, please. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Today's webinar is powered by your webinar, your ASPB membership. If you, for any of you who have not joined ASPB yet, you can join today and use the discount code presents10 to receive 10% discount on your ASPB membership registration. ASPB members get access to all other benefits, including this webinar, and all opportunities that ASPB provide are available at aspb.org for viewing. So you can check that out to learn more. Next slide, please. This webinar is recorded, and we have recorded a lot of other webinars. So if you're looking for additional plant science-related content in various kind of topic areas, feel free to check out our Plantae YouTube channel, where you can find many other exciting talks that we've done in the past. And as you explore and find anything valuable, definitely don't hesitate to share those resources and kind of help spread the content widely to share it with your peers, colleagues, and anybody in your network. Next slide, please. Before we get started, I'd like to go over some housekeeping items to make sure that you get the most of your attendance in today's webinar. If you are experiencing technical issues, please let us know about those by using the Q&A box, or you can email me directly at jpadilla@aspb.org. If you have questions for our guest speakers, please also add them in the Q&A box in Zoom, your Zoom panel. Our moderators will be working with the speakers to get them answered as well. If you have trouble connecting or need to leave the webinar early, as I mentioned, this webinar is being recorded and we'll post that once the content is processed and ready to go live. If you know a colleague or a friend or anybody within your network that was hoping to attend this webinar and wasn't able, feel free to share the recording of this webinar so that they could kind of catch all the great content that they missed. And so without further ado, I'm now going to turn over the floor to our moderator who will facilitate today's webinar. So with that said, take it away, Plantae Fellows.
Hi, hello, welcome everyone to today's Plantae webinar. My name is Nathaniel Oragbon, a doctoral researcher at CIPLAS, the Cluster of Excellence on Plant Sciences, Germany. And as already announced, the title of this webinar is The Role of Open Science and Data Sharing in Advancing Plant Science. Now, as many of us already know, the availability of scientific data is central to making research reproducible and enabling new discoveries. However, with the rapid advancement of multi-omics technologies, we're experiencing massive amount of data, and how we deposit assets and integrate this information will shape its usability for the broader community. Now, in today's session, we'll explore open science and data sharing in plant research from multiple perspectives, including research, teaching, data management, and entrepreneurship. Now, our panel comprises experts in molecular biology, bioinformatics, and data management who will share their experiences and insight with us. Now, by bringing this perspective together, our goal is to better understand the different expectations and needs within our community, and to highlight the potential and synergies that open science and data sharing can bring to advancing plant research. Now, as you already see, the webinar outline is shown here, where we're going to introduce our speakers and moderators. We have the talks, and then we have the panel discussion. And we already encourage you that as the talk goes on, if you already have your questions, you can drop them in the Q&A session. Next slide, please. Yeah, so for our speakers, we have Dr. Victoria Trakena, who is an academic expert in data science at CEPLAS, the Cluster of Excellence on Plant Science in Germany. We have Dr. Feijiang Fan, who is a postdoctoral fellow at the Chinese University of Hong Kong. Then we have a last speaker, who is Clay Lokshong, who is the genomic specialist at Hokkaido University, Japan. And then for our moderators, we have, on the next slide. Yeah, so for our moderators, we have Yishen Ko, who is a postdoctoral researcher at the Center for Soybean Research in Chinese University of Hong Kong. Then we also have a second moderator who is a postdoctoral researcher, Ved Prakash, Ohio State University. Then myself, postdoctoral researcher at the Cluster of Excellence on Plant Science, C+. And then we have the last, Krishna, who is a PhD student at the University of Southern Queensland, Australia. And so now, without further ado, we want to introduce our first speaker. But the next slide. Now I want to introduce the first speaker. We have Dr. Victoria Trakana, who holds a BSc in biotechnology and MSc and PhD in bioinformatics, all at the University of Wageningen, Netherlands. And his PhD focused on data analysis and software development for studying plant-associated microbial ecology and the role of suppressive soils against fungal plant pathogens. Then he also again continued as a postdoctoral researcher in microbial evolution, where he investigated the nuclear organization of the Verticillium dahliae genome. And currently he's an academic expert in data science and data management at CPLAS, the Cluster of Excellence on Plant Science, where he supports research, teaches and organizes workshops on data science and data management. And this is why we have him today. So now we give the floor to Dr. Victoria Trakana.
Hi, thank you for the invitation. Thank you, Nathaniel, for the introduction. I will now share my screen. Okay. If I get a thumbs up from somebody that everything is fine, then I can get started. Okay. Thank you for the invitation, as I said. Thank you for an introduction. My name is Vittorio Tracanna, as I was introduced. Today, I will discuss about data science. I will try to give you some ideas of how we do data science at CPLAS, the Cluster of Excellence in Plant Science in Germany. And maybe I will introduce you to a new tool that may facilitate your research. Okay, so first of all, I work at a research cluster. This means that I am part of a team that tries to support plant science research and data management, RDM, and it's trying to put together multiple institutions that try to manage and share the data effectively and together. So when you have different researchers that come from different universities and they work within the same cluster, it is very important to harmonize how this is done. Okay, so first of all, RDM, what is it? Research data management. It's like a series of concepts that revolve around the organization, storage, and preservation, and sharing of data. The idea is that it makes sure that the data is reproducible, that it can be easily shared within the consortium and outside, and not the least is that it actually complies with requirements from funders, from funding agencies. As any computer data science presentation, even related, here I have on the side of the screen an XKCD comic. It's mandatory. I don't make the rules, but it tries to explain how you are your first collaborator, first and last. And when you wrap up a project, you will be glad to yourself if you actually did the RDM from the beginning or earlier on during your publication, because 3 years down the line, you don't remember anymore where that PCR results are. Okay, so we work with in biology, and working in biology, we know that everything has a life cycle, including data. Here on the side, I have a very little and simple description of the different stages that of the life cycle of data. We start from the planning phase, then there is collection, processing, analyzing, preserve. Once you're done with your analysis, hopefully share it with the community, and even more, hopefully then this data can be then reused by others and a new plan can start and so forth. I don't need to go in details into these stages, but what we all have to agree and understand is that while it may seem so simple, it is not the case that a project lives just on its own. Oftentimes, and most of the time, what we have is projects cross-feeding on each other's. Whatever project you're working on, it's probably based on previous research, and therefore we want to be able to pass between one and the other as seamlessly as possible. How can that be ensured? It's part of my job and part of my responsibility, and it all starts here at the plan. In the moment that we agree on one plan on how to share, organize, and store and preserve data, then it becomes much simpler to jump from one to the other, from one project to the other, and then ensures that the research that you do, and that should be everybody's interest when you're doing research, is that whatever you are working on can then be reused and can be further be useful for the future generation of scientists, right? We are just standing on shoulders of giants. And by doing proper RDM, that's how we ensure that that's possible. Okay, when it comes to philosophy of research data management, one of the most classic examples is trying to do it with following the FAIR data principles. FAIR standing for findable, accessible, interoperable, and reusable. I will not try to go into detail into the concepts, but if you have to take home one message from my presentation today, and you want to go one step deeper into understanding better research data management, I can strongly recommend reading up the paper that originated these FAIR guiding principles that is listed here. It's a rare case in the field of research data management where actually the paper is accessible, easy to read, and understandable, and most researchers probably will agree with what is contained inside. Okay, but what is important for me, talking about FAIR data principles, is that fairness of data really is not only related to the data itself, but even more so about the metadata infrastructure. Just a brief introduction about what metadata is, in case some of you are not familiar. Simply put, metadata is data about data. It's all the accessory information that comes with your raw data or analyzed data that allow you to better contextualize and understand it. To give you an example, imagine that there is a website that it's called genome.com. We go on this website and then there is a list of ATGC, ATGC, ATGC. That's, I would say, arguably completely useless. Yes, it may be a genome, probably it is a genome, but I don't know who has done it, when they've done it, what technology did they use it. I have no reference point. However, if at that same website, the information that is listed there is arabidopsis-genome.com, and it's like, there is like who has performed the analysis, who has sequenced it, what technology did they use, what tool did they use to assemble it, where is the data stored, and all this kind of information, that suddenly, while the data is not physically there, it's a much more important source of information. Okay, but enough, unfortunately, about FAIR. Today, I will try to focus a lot, or focus my talk about ARC, Annotated Research Context. It is a framework that it's used within our cluster of excellence of plant science in Germany, and it tries to encapsulate all the components of your research project, including data, metadata, and analytical workflow, all in one bag. It promotes FAIR principles while it does not necessarily enforce them, but it helps the researchers to be fair with their data. It enhances data reproducibility and transparency, and it's a really good system to facilitate sharing this data with the data hub and in general with the other people that are part of this consortia and outside of the consortia, as I will show you later. Throughout this presentation, I may be using some terms that may not be super relevant for you, but just to be exhaustive, if I talk about data plan, that's a web-based tool for creating your data management plan. It's the white part in the research data lifecycle slide that I showed before. And it allows you to write a data management plan that fits your needs. Architect, it's a graphical user interface tool that helps you create and manage your arc, that you see that arc at the beginning and arc in Architect. It's essentially a software that allows you to create an arc and maintain it. And then there is the Plantae Data Hub, which is a cloud-based platform where you can upload your data and share it with the plant science community. Okay, annotated research context, ARC. It's your whole investigation data, metadata, accessory information, all of it, annotation, all of it inside of one box, of one bag. It can work as a single entry point where you can use it to, you can use your ARC to then share your data with other existing repositories, a specialized endpoint, which can be, for instance, ENA, a Pride archive, a bioimage, and so forth. Additionally, it is indexed and therefore can be searched using the relevant keywords where you can search for projects that may be relevant to your interest. Let's say I was working with Verticillium dahlia, you can type Verticillium dahlia and all the projects that contain that type of organism will be listed. And it also works as a platform where you can then get a DOI, publish your, just the research, it's not about publishing a paper, it's about publishing your research data with all the data and metadata. And then this can be also be used and be referenced inside of a publication to be complete, instead of having this very annoying data is available upon reasonable request type of flags at the end of a publication. You can provide your whole data and metadata in a very rich form. Okay, I've been talking about ARC, I've been saying it's your research in a box, but what does it exactly look like? The secret behind all of this is that ARC is essentially a predefined folder structure. We have our main folder that may be the name of your project, and then it will have a name. Then there are 4 different pre-generated subfolders: studies, essays, workflows, and runs. Studies and essay are related to the experimental data, while workflows and runs, as the name suggests, are related to computational workflow and analysis. And then inside of the main folder, there is also an ISA investigation Excel sheet, which is just an Excel sheet that contains information regarding to the publication, such as a DOI, if it's related to a publication, people that were involved in this research, or just a brief description of the project. As I said, inside of studies and essay, you will find the experimental data, and there are subfolders that are automatically created. For generally speaking, we prefer to keep inside of studies all the material that it's at the beginning of your investigation. Like, let's say if you're working in an experiment that has plants and infects them with fungi to see some effect, you may have inside of study, you will have a folder describing your plant material and another one describing your fungal material. It's all the starting material for your investigation. Can have all sorts of metadata information, such as a taxonomy ID, growing condition, also growing status that you got your plant from, material from. Was it a pre-germinated seedling? Was it a grown plant? Can be anything. Then inside of assay, we keep the experimental, all the parts, in assays, we keep all the assays that you can run inside of, for instance, inside of a lab or a greenhouse. So one in this case that we're describing an experiment of interaction between plant and fungi, there can be an infection assay that will be there. Maybe there is a harvesting assay where we harvest the plant and gather material. Then there can be some sugar quantification assay. All these different assays will be different folders with their own data set, protocol, and Excel sheet that describes the metadata. And same goes for workflows that run. I will not go into details there, but you get the idea. Okay, when it comes to metadata, a very important concept that we use inside of ARCs is to use ontology. Ontology, just to, as brief recap for those of you that may not be familiar with it, it's a construct that combines features of a dictionary, taxonomy, and thesaurus. So dictionary, as we know, it's just alphabetically lists terms and then their definition. So for instance, we have pizza. The definition is a dish made typically of flattened bread dough spread with a savory mixture, usually including tomatoes and cheese, and often other toppings, and baked. Good, we all can agree with this definition. But you can also have taxonomy related to, again, traditional Italian dishes. You can go from the left to the right. So you start with traditional Italian dishes, then you have macro categories of pasta and pizza, and then you can go even deeper looking at individual items. So taxonomy, it's this sort of sorted hierarchical list where the deeper you go, the more precise you get. And then thesaurus is this idea of having synonyms and relations between terms. So pizza is kind of a lammatron, which is kind of like a focaccia, which is kind of like a Flammkuchen. Okay, and ontology, the objective is to try to put all of them together in a graph-like manner. This can be the main objective of doing this is to model the relationship between the terms and make it machine understandable, so that so for instance, this search engine for different projects that I was mentioning before can be used to search this graph. Okay. Ontology is based on a semantic triple. We have a subject, we have a predicate, and an object. Going back to the pizza example, we can say margherita has topping tomato sauce. So once again, subject, predicate, object. Margherita has topping tomato sauce. But margherita also has topping mozzarella cheese. And you can see how this now creates some sort of graph. But we can also say margherita is type of pizza. So we can just, by changing the predicate, we can change the interaction between these two terms. These terms can be one directional or can be bidirectional, but in this case, once you change the direction, you also have to change the predicate. What we also can do then, we can create a whole menu, a knowledge graph of pizzas, where you can have pizza and then you have different types of pizza, different type of ingredients, and then you can try to using this network, you can try to ask questions. So I can ask, name all pizzas that have mushrooms as toppings, and at that point it will tell you, hey, there are two hits in the network, which is quattro stagioni and funghi. But why am I talking about pizzas and networks and so forth? I thought we were talking about plants. By the way, all of the things that I've described here exist in reality. You can go on the internet and look up the knowledge graph of pizza. It's actually quite funny that people have a lot of time on their hands and they can create any sort of knowledge graph. But why am I talking about this? This is because inside of ARC and inside of when I was saying that the metadata is the most important part about data, is that we can use this knowledge graph to flatten it and use a 2D table, just like an Excel sheet, which you may be very familiar with, to represent this type of interaction. So for instance, we can have, I don't know if you can see, but here on the left, we may have the subject. It's the first entry is called G2_UMV for 15 milliliter. Doesn't matter with this, it's just a subject. Then you may have a predicate, which here can be factor temperature. That's the predicate in the middle. So you have subject, the first entry. Predicate will be the column header, and then the object will be the value, which here is 30 degrees Celsius. Why is this important? Is that it allows you to replicate this 3D structure that we saw before into a 2D table. Yeah, so this makes it very easy to use the classic Excel sheet type of workflow that we may be familiar with, but enrich it and use an ontology that it's in the background, the tool that we use, Architect, that can create this, add this information to your metadata. In case that you are interested and I pique your interest about how to use this type of information, there is a getting started guide that you can use it's hosted at the data plant. You just have to Google data plant getting started, and then you can do it. It's a very simple guide. You should be able to complete it within 1 hour, 2 hours. You can always reach out to me or other members of data plant and ask for information. And from that point on, you are ready to use ARC and start your ARC journey. With that, I would like to just show you a very simple example of how this may look like. So once you're done creating your ARC, you can then share you're created your ARC. You have annotated all the metadata that is available to it, and then you can share it on this Plant Data Hub, which is the platform that we use to share the data. It's not limited to CEPLAS, it's not limited to Cologne or Germany. It's open to everybody that creates an account, and then you can upload your data there and share it with other members of the plant science community. Here we see different published articles. Everybody that logs in, you see I'm not logged in the upper corner. You can see all of the different projects that are listed there. There are plenty that are open already. And just to give you an idea of how an individual ARC will look like, I do not have the time to go into detail about all the parts that are important, but I have a small video. So this is inside of one of the ARCs that we saw on the previous page. You have a landing page with all kind of information regarding to the studies, the essay. You have an abstract, pictures. You can create it as you want. Then we can go into essays. And here we see there are different essays. And inside of one of them, here's the metadata. If we click inside of the data section, we will see that it has the relevant raw data that we can use. And the good thing is that once a person is familiar with the concept of ARC, it can always go to a new ARC and intuitively know where everything will be. And this is how we ensure reproducibility and ease of sharing the data inside of a cluster of excellence. With that, I would like to thank you for your time. If you're interested in ARC and its application in plant science, please feel free to reach out to me. And with this, I will be taking questions.
Thank you very much. We'll have our Q&A session after all the 3 presentations. Our next speaker is Dr. Kejin Fan. Dr. Fan obtained his PhD degree in molecular biotechnology from the Chinese University of Hong Kong. He is now a postdoctoral fellow at National Key Laboratory of Agrobiotechnology of the university. Dr. Fan's research focuses on non-coding RNAs and their regulation on soybean rhizobium interaction. Let's welcome Dr. Fan.
Hello everyone. Thanks for the invitation. I'm very happy to share my current research here. The topic is unlocking soybean nodule development's transformative power of RNA sequencing. Soybean is one of the most important oilseed crops. It contains 90% oil, 34% protein, and 21% fibers. It can be used as both food and feed. Soybean is widely planted around the world. America and Brazil have the largest soybean production. Soybean are a major global agricultural commodity with significant international trade. China is the world's largest importer of soybeans, with Brazil and American being the leading suppliers. This is why we study soybean in China. Soybean nodulation is a symbiotic relationship between the soybean with the soy bacteria rhizobium. It plays a critical role in the nitrogen cycle. These bacteria form nodules on soybean root, where they convert atmospheric nitrogen into the usable form for the plant, primary ammonia. This process, known as nitrogen fixation, it can reduce the need for nitrogen fertilizer in soybean production. Let's look more details about the nodulation. There are two kinds of nodules in legume. One is indeterminate nodule, such as the medicago. The other one is a determinate nodule, like soybean. So today we mainly talk about the determinate nodules. Rhizobiums can get into the soybean host by two ways. One is from the root crack, means crack entry. The other way is from the root hair through the T3SS secretion system. The soybean root hair can release the flavonoid to attract the rhizobiums. Rhizobiums can attach to the root hair and squeeze some effectors or knobs into the soybean host. These effectors and knobs can be identified by defense proteins triggering a defense response that inhibits nodulation. For example, the NOP, some kind of NOP can be identified by the RG2 and conquer the infection threat formation to start nodulation. So the host specific nodulation depends on the knobs secreted by T3SS or rhizobium. In our research, we used two dominant standard rhizobium from the alkali saline soils in northern China. Here we call them R4 and R2. R4 could form nodules both with a wild soybean W5 and a cultivated soybean CO8, but R2 can only form nodule with the wild soybean W5. W5 is a RG2 loss of function line, but CO8 is a functional line. Then we construct their genome and compare the NOP's proteins sequence among the different rhizobium. We found that only the NOP and NOP-I shows the sequence variation among the IFT1 restricted and non-restricted strains. So we suppose that NOP-I and NOP were responsible for the RG2 controlled nodule host specificity. Here we show that please wait. If the knobs are responsible for nodulation, the block of the T3SS system will recover the CO8 inoculation. So we construct the IHCN10.5 mutant. IHCN was a putative ATP synthesis involved in the secretion of knobs through the T3SS system. So we found if the T3SS system is broken, the knobs cannot be transported into the soybean. The R2 can form nodules with the CO8. We also confirm which knobs is responsible for the nodulation. So we replace the R2 NOP with R4 NOP, and we found that the R2 can form nodules with CO8. But if we change the R4 NOP with R2 NOP, this soybean cannot form nodules. So we prove that NOP is the determinant factor in host specificity and compatibility. To further study the NOP and NOP-I effect on soybean nodulation, we made different mutants by exchange the NOP and NOP-I between R2 and R4. We have this kind of strains as the R2 and R4 wild type. Also the R2 NOP exchange with R4 NOP, we call it R2P4. The R2 NOP-I exchange with R4 NOP-I, we call it R2R4. Also the T3SS mutant, we call it IHCN. Same with the R4, we have the R4P2 and the R4I2. We use these strains to inoculate the wild soybean W5. 28 days post-inoculation, we harvest the root and nodules and extract their RNA and send for the iron sequencing. After the iron sequencing, we think our data is pretty good, and so we back to check the phenotype changes. We found that these different strains didn't cause the significant changes in plant height, but they caused the nodule number changes, especially the NOP-I. Here you could find that the R2R4 compared to the R2 wild type, the nodule number is higher. So this means the R4's NOP-I could help to induce the nodule formation, but the R4I2 has lower nodule number compared to the R4 wild type. This means the R2 NOP-I could reduce the nodule number formation. Then we check is the NOP-I effect on root gene reprogramming. So we firstly identify the differential express gene and use this DE gene to do the KG analysis. We found that when R2, R4 compared to the R2 wild type, this differential express genes was significantly enriched in the isoflavonoid biosynthesis pathway. As I mentioned before, the isoflavonoid was released by the soybean root hair to attract rhizobians. So this will help to increase the nodule formation. And we get our conclusion here is R2, R4 induce the isoflavonoid biosynthesis, which could induce the nodule formation. Then we check the NOP-I and NOP effect on nodule gene reprogramming. At previous studies, people always think the NOP's proteins only works on the nodule formation, but we want to check this NOP's function on mature nodules or on nodule development process. So we also get the differential expression gene first and use them to do the KG analysis. We found when I2, I4 compared to the R2, the differential express genes were enriched in isoflavonoid biosynthesis, also the hormone GA and SA biosynthesis pathway. The same is the R4, I2 compared to the R4 wild type, the DE genes was also enriched the GA and the SA biosynthesis pathway. So we think NOP-I was related to the hormone GA and SA biosynthesis and metabolism. Now look at the NOP. When the R4P2 compared to the R4 wild type, the differential express genes were enriched in the isoflavonoid biosynthesis. Also, the GA cytokinin pathways also related to the defense-related MAPA signaling pathway. Then we check if we totally block the effectors and NOPs get into the soybean host, if there anything changes, this differential express genes show that the nodule's defense mechanism or senescence-related genes was changed after we block the T3SS system. So we're pretty sure that NOPs are not only related to the nodule formation, but also control the nodule development. As we found before, the NOP and I show strong effects on nodule hormone signaling, and the plant hormones is the key regulator of the nodule formation and nodule development, and show different distributions in the whole nodules. Just like this review showed that this data was drawn from previous studies, was not verified before. So we want to verify the hormones distribution in nodules. In previous studies, people always use the hormone antibody or use the hormone sensors to detect the hormone distribution. But we use the spatial metabolomic method to detect the hormone distribution because this method allowed us to detect different hormones in one nodule at the same time. This will help us to do some researches about the hormone crosstalk. Let's introduce some background knowledge about the soybean nodules. The soybean nodules have two kinds of cells. One is the infected cells. We also here at the central region, we call it symbiotic region. Also, the uninfected cells includes the epidermis, inner cortex, outer cortex, and vascular bundles. Here is some of our data about the spatial metabolomic data here. The blue color means there is no hormone or very low-level hormones. The red and the green color means that there's many high-level hormones here. So you could find the SA and the cytokinin are accumulated in the outer and the inner cortex regions. But the NOPS proteins was produced by rhizobium. It should be in the infected region, but the NOP changes will significantly regulate the SA and cytokinin biosynthesis pathway. So we began to think about will the NOP or NOP-I be transported into the uninfected zone to change the soybean host plant gene expression. But now we are still working on it. There are three common used spatial transcriptome method. One is the temporism, the other one is the STILL-seq, also the spatial single-cell transcriptome. Advanced technology make us to make it possible to get both gene transcription and localization informations. So I used the STILL-seq method to detect the gene distribution in the nodules. You could find this is some of my data. Here we use two nodules. And after the sequencing, we did the cell type clustering analysis. We totally get 12 different cell types. This cell type 5 is the first identified by us. This never identified before. And we found that the CK and SA accumulated in cluster 1, cluster 3, cluster 5, and cluster 6. So we want to confirm the genes related to the SA biosynthesis, signaling, transduction, or metabolism process genes distribution in the nodules. We found that the red, here is the red point is the site of gene detected. So we found that SA-related gene and the cytokine-related gene was significantly enriched in the cortex region or the outer layer of infected zone. So this is directly consistent with the hormones distribution. As NOPI and NOPI also showed an effect on the pathogen defense pathway and assisting the nodule senescence-related pathways. So we checked the plant pathogen defense genes distribution and senescence-related genes distribution. We found that this plant pathogen defense-related gene and senescence-related genes were all enriched in the inner cortex or outer cortex region. This is very interesting because before, people always think the nodule senescence started from the central zone of infected region. But now we found there is a strong effect about the senescence-related genes was in the uninfected zone. So we began to think about if the nodule senescence is also regulated by the plant host, not by the rhizobium. Now is my conclusion here. The knobs are key regulators of nodule organogenesis and subsequent development. Knobs may mediate the rhizobium effects on legume host genes transcription, especially genes involving hormone signaling. The spatial distribution of SA and CK correlates with the expression domains of their associated signaling pathway genes. Also, the last one important is dissemination of RNA sequencing data will foster progress across the research community. And now I will thank to my supervisor support, Professor Homilan, and also thank my teammates help in the bioinformatics analysis, Ms. Zhang Hui and Wang Liping, two PhD students. Okay, thank you.
Thank you very much. Our next speaker is Dr. Claire Yip Lok Chong. Dr. Chong obtained her PhD degree from the Chinese University of Hong Kong. She then moved to Japan for her postdoctoral research. She is now a genomic specialist at Hokkaido University in Japan and a senior agro data scientist at a startup company to bridge genomics and agriculture through AI. Dr. Zhong is an expert in genome analysis and data-driven agriculture. Let's welcome Dr. Zhong.
Thank you, Dr. Gu, so much for the introduction. So, yeah, well, but welcome all of you today. Hey, it's wonderful to have this seminar and have an audience from different parts of the world. So I will be sharing how open science and data sharing helps promote sustainable farming to support our future as mankind. Before we begin, let me introduce a bit about myself. So my name is Claire Zhong. I was born and raised in Hong Kong until graduating from my PhD in bioinformatics in the Chinese University of Hong Kong 3 years ago. So I moved to Hokkaido to continue on genome research and also provide sequencing service. And lately this year, I joined this from AI company in this field to work on smart agriculture, to analyze satellite images and soil chemistry data to help farmers and food companies to make the best evidence-based decisions in farming and supply chain in decisions. Outside my main work and study, I had the honor to participate in some IT conferences, which promotes the usage and sharing of technology and data. Through these experiences, I get exposed to the culture and benefits of openness from different perspectives across different research fields, beyond academia into different industries and across regions and countries. All my life, I've been surrounded by plants. I was born and grew up in a community with a huge garden, which is very special in Hong Kong as a metropolitan city. I then chose to attend this university known for being a hill city with lots of greenery. And now I'm in Hokkaido. Our university has a farm within the campus, and I enjoy driving in the farms in the suburb to pick fruits and relax with the scenery of vast fields of wheat, rice, and corn, etc. It makes me feel closer not just to the mother nature, but also to the origin of food that sustains my life and brings me so much joy to look forward to every day. But what exactly is our relationship with food crops? According to the World Bank data, global population is going to reach 10 billion in 2050. A research in 2021 analyzed hundreds of scenarios in 26 global studies and projected a 50% increase in food demand by then. We need to produce ever more food to feed everyone. On the other hand, food supply is being heavily challenged. We are experiencing drastic climate changes with ever more serious and frequent floods and droughts across the world. And freshwater availability is decreasing, yet we need around 70% of the global freshwater withdrawals done by agriculture, and we have shrinking arable land per person due to urbanization and land soil degradation. All of these mean that we need smarter agriculture to feed the future. So we need crops that can adapt to these tough and unpredictable environments, and data-driven technology-enabled ways to look ahead the environmental changes and make wise plans in response. So my journey began with soybean genome research during my PhD study under Professor Channing Fung, in partnership with the State Key Laboratory of Agrotechnology led by Professor Lam Han Ming. Soybean, as you heard, is a major soy seed and protein source for both humans and livestock. As a legume, it has a unique relationship with symbiotic bacteria that transform nitrogen from the air into biologically usable nutrition, enhancing soil fertility and reducing the need for chemical fertilizers. This not only benefits farmers, but also contributes to sustainable agriculture by lowering input costs and environmental impact. We want to understand the nature's designs for the soybean, effectively find suitable varieties that can adapt to the challenging environment, and maybe find ways to improve the crop. However, one of the big challenges was the lack of genomic resources for wild soybeans. Genome is the total hereditary information. A reference genome is like a map of the city. If the map is clear, we can find the right streets that are the target genes and build on it. If the map is blurry or missing rows, we get lost. That's why a quality genome is the starting point for comprehensive biological studies. At that time, the research field relied heavily on a single reference genome, Williams 82, which is a cultivated soybean variety. This reference was valuable, but it could not capture all the important genomic regions that are absent in that single variety. Moreover, it was built on short-read sequencing technologies, which limited the quality and completeness of the information. When the reference was more fragmented, it was difficult to study variation that span larger regions. That is where international collaboration and open data sharing played a crucial role. Multiple groups around the world come together and work to renew and improve their existing cultivar soybean genomes. Our team also led the initiative to establish a high-quality reference for the wild soybean. And comparative studies across different soybean varieties, including the wild species, highlights the importance of having diverse references. They reveal structural changes in the genome that underlie important agricultural traits. Let me show you some examples. By comparing wild and cultivated soybean samples from a diverse geographical origin, we found the genomic inversion in chromosome 8 that brings the parent gene close to the inhibition element, which turns off the coloring in most cultivated soybeans. That's why we have white but not dark tofu. We found the cultivated soybeans are selected for having fewer copies of a digestive enzyme inhibitor for better nutrition values. Also, further studies led to the discovery of the loss of the relevant DNA fragments in cultivated soybeans, which prevents the plants from growing too long or bending easily. When the plants are straighter and stouter, they can be harvested more easily. As we are sharing our findings on open access to the scientific community for research groups anywhere to use, scientists all over the world are making use of this reference to conduct their own research. And we see not 3 studies being published, and based on this reference, so discoveries are ever ongoing. Along our research, right from the very beginning, when deciding what soybeans to study and obtaining in the latest data, public databases such as Soybase in the US were very useful. And beyond the data, sharing of biological materials are also crucial in contributing to collaborative research. 5 years ago, I shared out an online database of the narrow gene bank project in Japan and reached out to them to request receipts. The team was very kind and efficient in responding and shipping us the materials. These resources make it possible to evaluate relevant varieties in international collaborative research. I'm glad to see that the research team and the body of research results at CHK continue to grow after my graduation. For instance, the team created and is maintaining the database called Wild Soy DB to share genomic resources focusing on wild soybeans. I also see that the CHK team has initiated the seed tech company to facilitate the preservation and improvement of vegetable crop farming and research, via data sharing, management of the storage and sharing of vegetable seeds, and also public science education. It's very much a system of give and take. Open science accelerates progress when we freely exchange data and materials. And besides, for give and take, it is not just giving back. Very often we add value and then feed forward the blessings we have. Meanwhile, open science in agriculture goes beyond laboratory research. To truly enable data-driven farming, we need to integrate many layers of information. Human activities such as sowing, watering, and harvesting, environmental factors like weather and soil composition, and are monitored via real-time satellite and sensors, all provide valuable insights. Together, these datasets help us better understand the crop growth and guide farming practices. And just like the listen field, our CEO will say, he wants to listen to what the field is telling us. And in practice, data-driven agriculture can provide tremendous value. For example, growth period prediction helps schedule manpower and resources, like when do we need how many people and tractors to harvest. Yield prediction supports food supply planning, and early warnings of drought, pests, and disease allow farmers to act before the damage is done or become severe. Optimization of watering and fertilization reduces waste and cost. All of these applications contribute to making farming more resilient and efficient. One of the most exciting aspects of my work today is bridging research with technology in industry. Open standards such as APIs and the fair data principles allow different systems to easily exchange data. And this makes it possible to quickly integrate various new technologies into practice. So we are applying the AI, the latest like LLM, and I'm also working on some prediction system. And for this view, actually he starts translating food information from satellite images into APIs and first started the business. And we are working to combine different bodies of analysis to provide high values to farmers and food companies. And in the industry, the focus is not just on novelty like academia, we want to publish new papers, but also on providing continuous, reliable services, maintaining close communication with stakeholders, and supporting continuous improvement cycles. This complements the innovation-driven outputs from academic research. And to make data-driven agriculture a reality, close communication with all stakeholders is essential. That's why we need to consider her different aspects and to integrate data from different perspectives, like biotech and bioinformatics factors collected from different time points and at different scales. We want to listen carefully to different feedbacks. And for this field, we also conduct like education sessions to the farmers to promote the acceptance and know-how of like using in the app we develop for the data-driven agriculture. And we want our farmers to understand about the sustainable farming practice. And the company also conduct collaboration with universities and provide internship opportunities to students for continuous growth and research. So to truly make an impact, I believe that we should bridge borders in multiple dimensions, to consider both biotech and abiotic factors, to connect between wet lab, dry lab, and field research, to collaborate between academia and industry, to work together from bench to field and across the supply chain to consumers. And importantly, across countries and cultures. And to achieve all of these, most importantly, it is for all of us to have an open mind, whatever her status we are. By fostering open science, collaboration, and integration, we can move forward sustainable farming practices and empower the next generation of farmers, researchers, and actually mankind. So I want to thank my supervisor, lab mates, and collaborators and colleagues all along my journey, and to all of you for listening today. So thank you very much, and I'm also open to questions.
Thank you very much. And here comes to the Q&A section. We have many questions, but due to time limitation, we may have one question to each presenter. So the first question is for Dr. Victoria. How can individual researchers or smaller labs contribute their data to the Plant Data Hub? Dr. Victoria.
Hi. Yeah, you can create just an account through Data Hub. You can log in with your ORC-ID, for instance. And once you do that, you can just upload the data. Following the tutorial, you can learn how to upload the data to the Data Hub. And you can also create a group for free for your own, even small research group. And everybody can then put the data together and then share it with the community once you are willing to. It's not necessarily about sharing the data when it's finished. You can also do it in the preparation of, let's say, a manuscript.
Yeah, thank you. And we have the second question to Dr. Kejin Fan. In terms of data sharing, how do you find the uniqueness of nodule research compared to other plant issues? Dr. Fan,
It's
please.
very, yes, the data for the nodule part is very hard to apply to the other other plants because they don't have the symbiotic relationship and it cannot form root nodules. But for the legumes like the chickpea, medicago, it can still work. Yeah.
And the last question to Dr. Chong. Comparing the research in the university and the company, are there any different considerations to share data or to use shared data? Dr. Chong, please.
Yes, surely there is some difference. Like especially in the company, he do sign the non-disclosure agreement, and so there are restriction and that we don't share everything. But I think it is not contradictory to open science, because like we want to have the incentive for something we work to prepare it to be ready for giving values to farmers and company, and we want to make sure things are ready before we share. And on academic, actually it's not like completely contradictory. We do share after we finish a manuscript, but we also share between collaborative teams. So I would say that like there is like more systematic restriction and in the data sharing restriction, but the principle is not that different. I feel that. Yeah.
Yeah, okay, thank you. So time is running out. If you have further questions, you may contact the speakers after the webinar. So we'll wrap up the webinar.
Yeah, so thank you all for joining us today. On behalf of Plantae and the American Society of Plant Biologists, I'd like to sincerely thank our wonderful speakers for sharing their expertise and insights, and our audience for your active participation and thoughtful questions. One clear message from today's webinar is that collaboration, openness, and knowledge sharing are key to advancing the plant sciences and creating real impact for our communities. I encourage all of you to take these discussions forward into your research, teaching, and networks. And also, please stay connected with Plantae for future events and resources to continue learning and collaborating. With that, I would like to once again express our appreciation to everyone involved in making this webinar a success. Thank you for being here, and we look forward to seeing you at our upcoming sessions. Have a great day ahead.
Thanks, everyone. And again, thank you to our speakers and to our Plantae fellows, and also to all the attendees. Hope we see you on the next one. And we'll forward all the other questions to the speakers so that we can get those answered. Thanks, everyone, and have a great rest of your week. See you all on the next one. Bye.
Thank you. Bye.
Thank you. Bye.